So there's 3 different theories. I'll put them up here. If you don't mind, I'll snag this just so I can look at it. That's cool. How did you buy that up to put on here?
Yeah.
I should have done that. I mean, I would like to have it on my iPad too. So when they were thinking, because we know like DNA would divide, right? And when we were first trying to figure this out, we didn't know how it would work. So a few different theories. We thought, okay, well, if this is our original DNA, right? And then it divides. At first we thought, well, this one will just make a copy of both strands. So it'll just make a copy. So you'll have 2 new ones, and then you'll have the same old ones, right?
Mm-hmm.
And that's known as the conservative model. So, conservative. And so that was one way that they thought they did it. They just copied the whole thing, and then it came, here's a whole new copy. Another way that they thought it happened was...
Semi-conservative.
Yeah. And do you remember how that one worked?
Yeah, so you get 1 of each side.
Yeah. So you have 1 right there, and 1 right there. And so each individual original strand went with a new copied one. And then there's the last one, and if you remember how that one was.
That
one was like weird.
Yeah, all mixed up.
Yeah. So you thought you'd have some right here, some, and then this one mixed right there, like that. And so it's all mixed up. That's the dispersive. So I'll just put dispersive and what was it? Semi. There we go. Okay. So when we have that, they weren't sure how, which one was the actual process. So what we ended up doing is they did this experiment. Basically, they had, this bacteria, and they only allowed, because if you look at a nucleus, like the little subunits that make up DNA, they use nitrogen, okay? So they said, well, let's have one of these made with an isotope, nitrogen-15, I think it was, and we'll be able to detect it. So it's like a tracer, and you could follow where it was. So that's what they did with all these first original models. So they got one of these, and the bacteria only could use the nitrogen-15, so they could follow it. And then after the bacteria did that, then they only provided normal nitrogen. So then they could trace the difference between, okay, whenever it starts dividing, all the new ones are only going to be made with normal nitrogen, right? Does that make sense so far? Are there any questions?
Okay,
you got it? Okay, so you're good. So if this was the original strand, they had bacteria and then only made DNA with the nitrogen-15.
Mm-hmm.
So it's different. You could follow it, Then, so it made, bacteria made this, like the parent set of, DNA.
Mm-hmm.
Then they provided it. They didn't allow normal nitrogen to be there, which is why I only used the nitrogen-15, right?
Mm-hmm.
Well, then after that, they only provided normal nitrogen. So this, right here, which was made with nitrogen-15, and then when it's going to make new stuff, however it does it, it's only going to be able to use normal nitrogen. So, actually, I'll put that right there. It's only going to be able to use normal nitrogen. So, you can start seeing a difference. If it's really trying to classify so we can identify the new copied strands, and in whichever form they're going to happen here when we run the experiment, versus the original ones. So we can just keep our eye on the original ones, because they're nitrogen-15, and everything else that comes after them is going to be with the normal nitrogen. And so is that kind of...
So why did they want to do that?
So they want to do that so they could track, so they would watch how this would divide almost. And then if they could keep track of the original stuff, well, if the original stuff was all together like this, and then all the new stuff was like this, then you knew it was going to be this model. But if you had a mixture, you knew you could follow the new stuff and it was with half of the old stuff and the same here, then you knew it would be like this theory.
Okay.
And if it was like this each time, then you'd be able to follow it too. Is that kind of...
So they were just trying to find which one it went through?
Yeah.
For this person.
Perfect, yeah. They just want to know what happens with this original strand when you duplicate. Does it stay? Do they stay together and then just you have all new ones, or do you have one of the original strands goes with a new copied one, or do they just all mix up? So they needed a way to follow the old strands to separate them from the new strands and to see, how does this come apart and how do they duplicate?
Okay.
So, and that's why they used the isotope nitrogen-15, because it's still nitrogen. So electrically, the electron cloud, remember when we talked about, and I think it was like Bio 180, electrons determine how it reacts with other atoms.
Mm-hmm.
So the electron cloud's still the same. So it's still going to react. Just now it's a little different so we can follow. So they did this. And the first time it divided, it actually ended up being like this model right here. And what they did was they put it into a test tube. And the thing with nitrogen 15